Hello students, welcome to Vedantu Neet English. I am Ms. Gopika, your biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Please let me know in the chats if I am absolutely audible and visible, and the presentation also is beautifully visible to all of you. Yes students, quickly let me know how is the Josh on Monday morning. Okay, I am doing great. How are you all doing? Tell me about how all of you are doing. That is more important, right? Yes, I think we are live. Yes, we are live, right? No problem. Tell fast. Okay. Yes, we are live. Okay. Students, quickly share the link with your friends and ask them to join as well, and fill the chat box with fire emoji. Let's activate our energy for Monday morning, because today we are going to complete excretory product and the elimination part 2. So as of now we have completed a lot of syllabus. That is, we have completed mainly in your human physiology. We started with breathing, we are done with it. Body fluids and circulation, done. Excretory product and elimination is going to get over today. And I also finished sexual reproduction in flowering plants, molecular basis of inheritance, major chunk. Only HGP and fingerprinting is left. So yes, we have completed really high weightage chapters. We have been completing a lot, right? So yes, quickly share the link with your friends, and I'm so happy to see a lot of my DNA, RNA, proteins, all the students together here. Awesome. So let's start the class, students. Today we're going to do excretory product and elimination lecture 2. So what did we do in the last class? Mainly last class we understood what are the excretory organs, right? So I'll give you a quick recap of what just happened last class, so you'll be able to understand. So we understood different types of excretory organs depending on their habitat and level of organization, and mainly 3 nitrogenous bases there: ammonia, urea, uric acid. If an organism is giving out ammonia, it's called as ammonotelic. If an organism is giving out urea, it's called as ureotelic. If an organism is giving out uric acid, it's called uricotelic. And depending on their habitat, they will be classified on what I want to secrete, right? That is your types of nitrogenous waste. And next thing that we studied was human excretory organ, or your human excretory system, where we studied 2 kidneys, ureter, urinary bladder, their muscles that are present which will help them in contraction, peristaltic movement. All this we studied in human excretory organ. And also we saw the transection of your kidney, where you saw the cortex, medulla, where the nephron is present, where is medullary pyramids, renal papillae, renal pelvis, ureter, then your what else? You also saw the renal envelope. All this you saw. Then students, yes, kidney we did. Nephrons, your main portion. Nephrons is divided into 2, that is your glomerulus and your renal tubules. Under your renal tubules you saw PCT, DCT, loop of Henle, and you saw collecting duct, but collecting duct, many nephrons merge into it. And in your glomerulus you saw glomerulus plus your Bowman's capsule together will be called as Malpighian tubules. So this is what we saw in the entire nephron. So today we are going to understand why this nephron is there and what are they exactly going to do. Right, exactly going to do. And then students, we saw JG apparatus, that is whenever there is a problem with your GFR, that is your glomerular filtrate rate, okay, immediately there will be a entire mechanism who does the quality checking. So once the quality check is done, they will release renin, and that renin is going to continue the mechanism called as RAS mechanism that we are going to understand today. So yes, my rapid fire is over. I hope all of you are able to remember what we did last class. Now this class we are going to do mechanism of urine formation. I could not write full. Okay, ultrafiltration. That is again glomerular filtration itself. Function of all the tubules, mechanisms, that is RAS mechanism, ADH mechanism, ANF mechanism, countercurrent mechanism, disorders. This is what we are going to do, because I told it in one breath. My speed is not able to, I'm not able to control my speed. Chalo. So students, this was the tubule that I drew last class, so I took a snip of it, right? Yes, Hema, same for me. JJ is my favorite topic. So I hope all of you marked your attendance. If you haven't marked your attendance, quickly smash the like button, and if you're new to the channel, do not forget to subscribe, okay? Because you're getting physics, chemistry, botany, zoology, everything in one channel, including your practice papers, question papers, everything is in this channel. So why aren't you thinking of subscribing? So students, this is your tubule. You have your glomerulus plus Bowman's capsule, then you have your PCT, then your loop of Henle, then DCT, then collecting duct. This is what we are going to study. Today we are going to understand what these tubules are exactly going to do. How are they going to do proper urine formation, right? Are you all ready? Are you all ready? So yes students, mechanism of urine formation. First of all, understand, for urine formation to happen, there are 3 major steps. I know all of you know the major steps. Okay, that will be the first thing that you people would have studied. Ma'am, nothing else I study also, I will study the steps. Okay. I think, yeah. Is it straight? Yes. So students, today we are going to understand urine formation. The first ever step of urine formation is going to be glomerular filtration. Glomerular filtration. Second step is going to be tubular reabsorption. Tubular reabsorption. Third one is going to be tubular secretion. So these three steps mainly contribute in forming your urine. Now all of you just look at this and tell me where do you think the locations will be? Like, where is glomerular filtration happening? Where is tubular reabsorption happening? Where is tubular secretion happening? Just look at this and just tell me, ma'am, I can just look here and understand that glomerular filtration is happening obviously in the glomerulus. Okay, it is obviously happening in the glomerulus. Yes, ma'am, it's happening in the glomerulus. What about tubular reabsorption, tubular secretion? Very simple. Tubular reabsorption, tubular secretion is definitely going to happen in the renal tubules. In the renal tubules. Does anyone have a doubt in this? Just by looking at the name, right? Just by looking at the name, you will be able to tell where the absorption is happening. Okay. Now, students, understand your entire body. Lot of filtrate, okay. I'll just tell the number, 180 liter per day. That much, okay, that much of filtrate is going through these tubules. But how much urine are you passing it out? You are only passing 1 to 1.5 liters of urine. So do you see, around 99.16% is reabsorbed. So today you are going to understand why reabsorption is happening. You're going to understand what is exactly getting reabsorbed. This is your main task for the day, okay. So students, these three steps are there: glomerular filtration, tubular reabsorption, okay. So this is happening in the glomerulus. Tubular reabsorption, this will happen between the blood vessels and the tubules, and tubular secretions. Today in the class, you are going to actually see the diagrams. I have drawn the diagrams and you are going to see how this exactly happens. Okay, how this exactly happens. Okay, Siraj Deen, okay, thank you so much, dear. Okay, now let's get back to the class. Now students, look here, ultrafiltration or your glomerular filtration in other words. Now please do understand, this is going to happen. Now let's say I drew a glomerulus. Okay, let's draw this. Let's draw one kutti one, okay. We're not going to focus a lot on this one. Okay, this is how your glomerulus will look. This is how, I mean, the Bowman's capsule will look. This is how glomerulus will look. Right. Now understand, the first ever step called as the glomerular filtration will happen through these people, okay. So it is basically this people, okay, that is your glomerulus and Bowman's capsule, through with this only the filtrate is going to pass. Now, when filtrate is passing, it is normal for you to understand that if a filtrate has to pass, it will pass through different layers. Now you have to understand which are the three layers that is actually going to help you in your ultrafiltration or glomerular filtration. Why are you calling glomerular filtration and ultrafiltration and giving us double work? Simple thing, students. It is because in your glomerular filtration, everything gets, filtered out except your large proteins. Okay, so plasma gets filtered out without your proteins. That means mainly what is getting filtered out? Water, right? So through this you should understand that water is getting filtered, all the nutrients. Just think of what I taught you in body fluids and circulation. Think of the composition of plasma, nutrients, all the minerals. All these are getting filtered out through your glomerulus except for your protein. Okay, except for your protein. So I can write plasma minus proteins. Okay, plasma minus proteins. Plasma is there, but I'm minusing proteins. Understand, with proteins, your RBC, WBC, platelets, those people also are not going to go. Okay, so if you want to ask, ma'am, tell me everything about blood, like if blood is getting filtered, what and all will be lost? Understand, blood corpuscles will not go out. Okay, plasma without protein. Exactly, plasma without protein. So plasma is going out, but without proteins, RBC, WBC. In plasma, first of all, these are not there. But blood as a whole component, understand, these things will not get filtered out. Because these things are not getting filtered out, I will call it ultrafiltration. That means, understand that all these are getting filtered out. Ma'am, such a nice filtration, very nice, we like it. So that's why we call it ultrafiltration. Now understand, when you make chai, right, when you make tea, normal tea, okay, what do? Your mom takes one sieve and she pours this tea. What happens? If it is tea leaf, that will also get sorted. If it is those powder thing, that will also get sorted. So all this will get filtered out, right? Same way, understand, for your glomerular filtration to happen, there are mainly 3 layers. Okay, there are mainly 3 layers. Students, I'll just rub this and I'll write it down. Okay, 3 layers. Which are the 3 layers for your filtration to happen? All of you tell me in the chats. I know many of you know it. So here I'm writing. Without proteins. All of you in the chats, which are the 3 layers? No, Ishwar, this is a pure English channel. There are a lot of Hindi channels, no? Yes. Yes, larger proteins will get stuck. Okay, proteins will get stuck. So students, this is understood. Now I told you, for your glomerulus, that is for filtration to happen, 3 layers are there. The first layer, okay, these 3 layers are there. Just look here, I have drawn few things. First layer is going to be the endothelium, okay, endothelium of capillaries. This is capillaries, okay, you are supposed to look at this diagram and understand, okay, blood is going this way, so it will go through this. Second layer is going to be your basement membrane, which is a non-cellular layer, okay, basement membrane, a non-cellular layer. This downwards, right here, who is there? Bowman's capsule is there. So epithelium of Bowman's capsule. Epithelium of Bowman's capsule. Ma'am, then what have you drawn here, ma'am? We are not understanding. Let's try to understand that. Okay, look here, students. First I do endothelium of capillaries. So now if I draw a larger image of this also, you can just imagine, no, larger image of that. The capillaries will be like this, right? The capillaries will be like this. Afferent will be there, efferent will be there. Inside this afferent and efferent, if I draw the cells, right? If I draw the cells like this, these cells will be called as the endothelium of the capillaries. So I've separated those cells and I've drawn it here. Okay, that cells are there. Okay, capillaries, ma'am, understood. Now after the capillaries, there is going to be a basement membrane. Obviously, understand, in body there will not be just empty spaces like that. Between your glomerulus and your, glomerulus and your Bowman's capsule, there's going to be a space. That space will be called as your basement membrane. So this basement membrane is a non-cellular membrane. Understand, it's a non-cellular layer. Okay, this basement membrane is there, students. So understand, from here till the capillaries, from here basement membrane, after the basement membrane it will definitely go to Bowman's capsule. Ma'am, but you have drawn it like this. That means, understand, in Bowman's capsule there is a specialized cells called as podocytes. Podocytes, okay. So there is a specialized cell called podocytes. Now the students understand, this podocytes are arranged in such a way, like an intricate manner, okay. It could be like an interdigitating, we can say intricate manner. This intricate manner will allow spaces in between. Can you see spaces in between my hand? Yes, ma'am, we can see your face, right? You can see my face. So this kind of intricate manner, this podocytes will be arranged. Why they are arranged like this? To give a small space. Otherwise, how are you going to filter out something, right? That small space. So podocytes are arranged in such an intricate manner to give some space, and that space will be called as your slit pore or filtration slit. Slit pore or your filtration slit. Filtration slit. Students, I don't think you people shared the link with your friends. Okay, no one is doing anything. No one shared link, no one is doing anything. Let me wait. Yes. Share the link, all if you don't tell me, ma'am, no friends and all that, okay? No friends and all that. So students, see, these are the podocytes, and between the podocytes there are slit pores. You'll also see the word fenestra. Fenestra also is the gap, okay? Gap. But then you don't have to know that, because again, NCERT that much detail is not there, okay? You can just know podocytes. Yes, fenestra is the gap between your squamous epithelium. So these are podocytes, the pink color one which I have drawn. Between these there are spaces, those are called as slit pores or filtration slits. So now you can imagine that, okay ma'am, filtrate is coming, okay? This filtrate is passing through this, okay? Filtrate is passing through this, it is moving, right? It is moving and it is going out. Now where is it going to go? It is going to go into the PCT, correct? Always remember, from the Bowman's capsule it's going to continue in the PCT, from PCT to loop of Henle to DCT, then to the collecting duct. This is basically urine formation. So did you all understand glomerular filtration? So students, understand that, okay, there is one, this one, okay, glomerulus is there, Bowman's capsule is there, 3 layers are there. What are the 3 layers? Endothelium of the capillaries, basement membrane, epithelium of the Bowman's capsule, or you can tell podocytes of the Bowman's capsule. If you get confused with epithelium and endothelium, you can definitely tell podocyte of the Bowman's capsule. Between the podocytes, there are small spaces called as filtration slit or slit pores. Okay, hi, yes, hi, yes. Okay, now students, understand, when something is moving, right, when something is moving from one place to another place, definitely there's a pressure. And you can just imagine capillaries. See, this is your afferent, okay, this is your capillaries. Now I've never drawn it like this for you, but say this is your capillaries. From capillaries, say efferent is there, okay, afferent arterioles is there, same way capillaries is there, from capillaries efferent arterioles is there. Just looking at this diagram, what do you understand? Ma'am, I understand that here there is a larger area, right? In the afferent arterioles there is a larger area. This is a larger surface, yes or no students? See, more space is there for the blood to come. See, blood is coming like this, it's coming happily. Now when it comes here, in the small capillaries which are present in the glomerulus, understand, glomerulus is a tuft of capillaries. Now there, the diameter is going to be small, so the blood is going to experience a lot of pressure. Blood is going to experience a lot of pressure. Because of that pressure, they will be able to move through these 3 layers. Okay, always remember like this. When something has to pass from one membrane to another membrane, always there should be some pressure difference, right? So this will push, or this will experience a pressure, and because of the pressure, they will pass through all these 3 layers. Now, I told you that how much glomerulus is made. So you have to know that GFR, okay, GFR is your glomerular filtrate rate. Okay, that is how much filtrate is going out. All of you are following so far with me? Any doubts on this? Since I cannot see your chat, wait 1 second. Okay, now I can see your chats. How many of you have been marked your attendance? Mark your attendance. Mark Madi Jaldi. Yes. Okay. Yes. So students, look here. GFR is your glomerular filtrate rate. That is the amount of filtrate. Okay, the amount of filtrate that is coming out per minute. Okay, that is formed per minute or per day, depending on what calculation we want to take. So this GFR is glomerular filtrate rate. Yes, ma'am, understood. How much GFR is made, students? What did I tell you? Around, a healthy person will have around 125 ml per minute. Okay, per minute. Or in other words, an easy way to tell is 180 liters per day. Okay, 180 liters per day. Now, do you think this entire 180 liters you are giving out? No, ma'am, I am not giving out. I am giving out only 1 to 1.5 liters. So, even though GFR is this much, your urine, right, that will be given out will be 1 to 1.5 liters. That means a total of 99.16% reabsorption is going to happen. Reabsorption is going to happen. Is this clear or not, all of you? Is this clear? Okay, around 19 to 99% reabsorption will happen. because you can just look at the numbers. You can just look at the numbers. Here it is 180 liter. We are giving out only 1 to 1.5 liters. So obviously the rest everything is getting reabsorbed, right? Rest everything is getting reabsorbed. So I can say around 99% is getting reabsorbed. Okay, there's a calculation for this. You don't have to know 178 point that you don't have to know, but just understand this. Now, ma'am, how much blood is actually pumped? How much blood is actually pumped? Understand, 1/5 of your entire cardiac output blood, right? 1/5 of your blood is getting pumped. That means around 1100 to 1200 ml is getting pumped. That is equal to 1/5. How much was your cardiac output? How did we calculate the cardiac output? Stroke volume into heartbeat. That was around 70 into 72. We got around 5040 ml. So I will round off it as 5 liters. In that 5 liters, around 1100 to 1200 is going for filtration. So that means 1/5 of your total cardiac output. Are you all agreeing with me or disagreeing? Okay, if you disagree, then you should tell me the reason. Yes or no? No one is disagreeing. Okay, very good. So that means around 1000. So here I am writing, okay, around 1100 to 1200 ml of blood, okay, is filtered. And this number is given in your NCERT, okay. They have given that is around 1/5 of the total cardiac output. Cardiac output. Anyone has a doubt in your cardiac output calculation? Check body fluids and circulation chapter. Okay, body fluids and circulation chapter. Okay, ma'am, this is also clear. Now, ma'am, you told some pressure and all that, right? Students, actually, you don't have to study pressure in depth. Previous classes I have taught, but we don't see much questions out of it. But I'll just tell you the name of all these pressures, right? Okay, in case anytime it comes, we can't predict. So, we will just learn the names of these, which are the different types of pressure because of which the glomerular filtration is happening. Okay, glomerular filtration is happening. So the first ever, students always understand there is always going to be positive and negative people, no? Same way there is going to be positive and negative pressure in everything. In life also there will be a positive movement, there will be a negative movement. Many of them are not attending class thinking that okay fine, I will just give it up or I will study by myself I will do something. Understand, myself, nothing will happen. You need to have that motivation, you need to have that thing, okay, I will sit in the class. Now Madhumati or say Hasini or Soni or Madhu Krishna, Jenny, Lisa, Rani, all of these people sit throughout the class. Now their motivation is different from a person who is sitting and telling that I will do by myself without having a proper schedule. So understand, ups and downs are part of life, but you should know when to pick up your pace and just get back to the class and get back to studying. And this kind of crowd is actually disappointing, you know, when the teacher takes a lot of effort, comes here, teaches, and then you people don't show even half of it back. Actually, the teacher feels bad. Don't think that, oh, she's a YouTube teacher, she will not have any emotions, she just stand and teach. No, we care about the comments, we care about the likes, we care about how many students are joining us every day, how many of them are getting helped because of that. That matters to us. Okay, so like that only, students, everything, even in your glomerulus to move, there is a positive and a negative filtration. Okay, positive and a negative filtration. Give me 1 minute. Okay. Yes. So the positive, okay, the positive filtration is going to be your glomerular hydrostatic pressure. Hydrostatic pressure. I'll teach you very little about this, okay? I will not teach you a lot, like how I used to teach you before. I will not teach you because we don't see much questions, so we'll just do how much is required. Next, the negative pressure. Okay, this is the positive pressure. That is, this is the person who is telling, go, move forward, do it. Okay, like me. I'm telling you, just students, you have to do it, you can do it. So I'm the positive pressure. Negative pressure is relatives, some people who tell you cannot do it. Yeah, leave this. This is not your cup of tea. Neet is for people who are toppers. Nothing like that. Average students only become doctors. Toppers are not, toppers are all gone for something else, some other courses. The average students itself study very hard and they become Neet toppers. Okay, no one becomes a topper and then become a Neet topper. It is very different from school. Okay, so understand, this glomerular hydrostatic pressure is the positive pressure who is telling filtrate, please move from the glomerulus. Okay, now the negative pressure is going to be the ones who are going to stop this person. That is going to be your blood colloidal osmotic pressure. Okay, osmotic pressure. This is BCOP, and capsular hydrostatic pressure. Okay, hydrostatic pressure, CHP. Okay, CHP. Now students, as I told you, one is positive, one is negative. Yes ma'am, you already told us this. Now we have to do a small kuti calculation. At what pressure? Students, power has gone. Students, I am boiling here. Wait 1 second. I hope it works. Work. Okay, it's telling me to boil. It told me just boil, no problem. Okay, yes, 1 second. Yeah, okay, are you all with following? Okay, now students, understand this BCOP pressure is the pressure exerted because of the difference in the diameter in the afferent and efferent arterioles. Like I told you, now here if the tell the diameter is more, okay, if the diameter is more blood will come. More blood will come. Yes or no? So GFR will automatically increase. Now say here the diameter is reduced. So the blood that is going out is going to be less and the blood that is inflowing will be more. So automatically GF will be more. Yes or no? More you give to your GF, the GF is going to be happy. More you tell the GF, no, I will not come, I will do this, more the chance of GF to leave. Okay, so your GF, more the blood comes in, more the GF. Okay, more the happiness of the GF. Now here it is less. So chances of going out will be very less. That means the blood which is going out is going to be less. Okay, understood? So glomerular hydrostatic pressure is mainly because of the difference in the diameter. Okay, ma'am, understood. Now how much is it? It's around 55 to 60 mm Hg. Okay, 55 to 60 mm Hg. Yes, exactly. All of you listen to the class. What are you people talking? Okay, go to someone, students, as if WhatsApp does not exist. Go to WhatsApp and talk. Okay. Tell Divya. So students, understand, this is mainly glomerular hydrostatic pressure is mainly because of the difference in the diameter, and it is around 60 mm Hg. Okay. Now blood colloidal osmotic pressure. Blood colloidal osmotic pressure means we understood that when we were filtering out also some of the proteins are left there, like albumin. We study, albumin, fibrinogen, all this. So this albumin mainly what it will do is it will stop the water. It will put a negative pressure. Say imagine water is coming in. There's a big rock. Okay, imagine water is coming in. There's a big rock. Will water be able to move? Water will not be able to move, right? Because the rock is there. Same way, this albumin will try to avoid the water to come in. Okay, water to come in. So this is giving a negative pressure for the GF. When say she is trying to come in, your friend is telling don't come in. So what is happening actually? This friend is giving a negative pressure from her to come in. So your GFR automatically drops. So that is a negative pressure. Blood colloidal pressure, okay, blood colloidal pressure is around 30 mm Hg. Okay, 30 mm Hg. It is caused because of colloid, okay, colloid osmotic pressure. This is mainly done to make an osmotic balance. You're not studying, your friend is very concerned, so your friend is like, you don't need all this, okay, don't need all this, you shut up and study. So that is a good, in the sense, both are done to do an osmotic balance. Both are done to do an osmotic balance. So 30 mm Hg. Then you have capillary hydrostatic pressure. Student, this pressure is caused because, see, inside already one GF is there. Will you allow one more GF to come? If you try to allow one more GF to come, this both will start fighting, right? So the GFR or the GF, glomerular filtrate, which is inside, will be like, I am already here. Why are you bringing more people inside? I will not allow. So this will try to push and not allow the new GF to come in. Okay, very related to your life. Okay, so new GF to come in. So this will cause a negative pressure, that is called as capsular hydrostatic pressure. Clear? This capsular hydrostatic pressure is around 20 mm Hg. 20 mm Hg. So students, now understand positive, negative, they are going to minus and see how much is going to happen. So 60 minus 30 plus 20 mmHg. Okay, this is going to be your net filtration pressure, or even you can tell it net effective filtration pressure also. Okay, yes, is this clear? All of you please tell me. Yes, CHP is 20 mmHg. Yes, Hema, 20 only, I don't know. Yes, 20 and 30. Clear or not? First tell me this if it's clear or not, or else I have to explain again. Clear? Okay. So net filtration pressure is equal to, students, wait, I'll write this formula once again. They'll ask you, okay, they'll ask you question, net filtration pressure, how much is it? They'll give you 4 options. You should be able to calculate and tell. So this is how you will calculate. Net filtration pressure is going to be, okay, it's going to be GHP, okay, glomerular filtration hydrostatic pressure, minus BCOP plus CHP, because I am adding both the negatives, I am adding both the negative pressures and I am going to minus. GHP is around 60. Students, this can be 55 to 60, okay, it ranged from 55 to 60. 60 minus BCOP is 30, okay, 30 plus 20, that is going to make a total of 60 minus 50 is equal to 10 mmHg. So students, the net filtration rate or the net filtration pressure for the GFR, okay, for the glomerular filtrate to move inside through your epithelium, endothelium, basement membrane is 10 mmHg. Understood, all of you? This much pressure is enough. With this much pressure, they will try to move inside through the 3 layers, okay? They will try to move inside through the 3 layers. Is this clear? Yes, ma'am, it's clear. So students, what did we study? This is a very important maths, okay? This question will come. They will ask you, is it 1/5, 1/6 of your cardiac output? So you should be in a position to tell. And if any student has a doubt, understand your cardiac output is stroke volume into heart rate, okay? Stroke volume into heart rate, that is going to be equal to 70 into 72, makes an approximate of 5040 ml, or simply write 5 liters, okay? Simply write 5 liters. Done? All of you clear? Yes, formula and all you have. See, I have to teach formula. As a biology teacher, what and all I have to teach. Is this clear? Can I move on? We are going to study the next one, next topic. Yes or no? Yes or no? Tell fast, fast. Any doubts, ask me right away. Okay, right now ask me. So much, someone has so much to laugh. Who is that? Yeah, ma'am, teaching maths. Biggest comedy, no? Okay, did you all mark your attendance? Yes or no? Okay, I'll continue. Next topic. Wow, ma'am, what have you drawn? Glomerular, no, tubular reabsorption. So students, from the glomerulus, now the filtrate we pushed it with the pressure, okay, we only found the pressure 10 mmHg, the pressure, with the pressure it has come down, okay, it has come down through the 3 layers, endothelium, basement membrane, epithelium. Okay, ma'am, understood. Now it is automatically going to the PCT. Yes, automatically it has to go to the PCT. From PCT, it has to go to loop of Henle to DCT, DCT to collecting duct. Now we are going to the next one, that is called as tubular reabsorption. That means whatever is not required, okay, whatever is not required, this blood will reabsorb it. The blood will reabsorb it. After that, if the blood loses its pH or the osmotic balance, blood will give it back. So this is called as tubular reabsorption, means from the tubules we are reabsorbing whatever is not required. So all of you look here, this is your tubules, right? This is your tubules, your Bowman's capsule, PCT, DCT, loop of Henle is there, okay? Now we have to understand tubular reabsorption means around 99%, okay? Almost 99% reabsorption will happen in the tubules, okay? Reabsorption will happen in renal tubules. Agreed, all of you? In glomerulus, did I tell anything like this? Did I tell any of this dialogues? Yes, ma'am? Yes, all of you, right? Okay, very good. Now students, understand, every time the tubules will not do active reabsorption or passive reabsorption. They will do both of it. So during the tubular reabsorption, okay, during tubular reabsorption, they will reabsorb, okay, tubular reabsorption, they will reabsorb both passively and actively depending on segment to segment. I will show you depending on the material that they are going to reabsorb. They will do it passively or they will do it actively. Got it? Now let's see which will be actively taken, which will be passively taken. Now all of you know this. All of you give it a guess. Yes, Hasini, Tanuja, all of you give it a guess. Divya. Okay, I'll only tell it. Now very simple, students. When I want to reabsorb, let's say I want to reabsorb glucose, I want to reabsorb amino acids, okay, amino acids. When I want to reabsorb glucose, amino acid, potassium ions, when I'm doing this, I will try to do active reabsorption. Agreed, all of you? Yes or no? Yes, ma'am, agreed. Okay. So when I want to do glucose, amino acids, or say, let's say sodium ions, I will try to do active reabsorption. Okay. I will try to do active reabsorption. Active. But at the same time, imagine I want to do absorption of nitrogenous waste. I will not waste my energy. I'll do passive, right? So when I have to do nitrogenous waste will definitely do passive, right? Passive. Now what about water? Water being the most important, what about water? Students, water in the initial segments, okay, initial segments of the PCT, DCT, it will be reabsorbed passively. But later, when we have to do conditional reabsorption, we will do with energy, that is active reabsorption. Depending on if body's GFR rate is here and there, some problem is there, that time we will do conditional reabsorption. That means in the initial segments, water will be reabsorbed passively. In the later segments, it can go actively. Clear? Can go actively. So is this clear? So 99% of reabsorption will happen in the renal tubules. Tubular reabsorption can happen two ways, passively or actively, with energy or without energy. Glucose, amino acids, sodium ions, I will reabsorb actively. Nitrogenous waste passively, because it's anyway waste, right? We don't have to take tension. Now, so what is going to happen from the tubules, okay, from the tubules, all this is going to get reabsorbed. Okay, all this is going to be getting reabsorbed. Let's say the orange dot is Na, okay, Na plus. Let's say the next one is amino acid, the next one is glucose. Okay, all this is getting reabsorbed from the tubules. Are you all understanding? Okay, they are all getting reabsorbed. Reabsorbed. What is this? What I've drawn? This is your capillaries. Okay, this is your capillaries. Yes. So, very simple, from the tubules, whatever we have needed, like whatever is needed for us, we are going to reabsorb, and those reabsorbed things is going to be in the capillaries. Okay, capillaries. Yes, fine, done. Okay, now next is tubular, okay, tubular secretion. Ma'am, oh, reabsorption understood? Okay, reabsorption understood? Okay, fine. Now what is secretion, ma'am? Okay, secretion, very simple. Reabsorption, I took everything I wanted. Now I understood that, okay, because I took everything I wanted, somewhere there is a problem with the ionic balance. Okay, somewhere there is a small issue. So when the blood understands that there is a break of ionic balance, the blood will give back that things that are not necessary. Are you able to imagine? One side reabsorbed everything to the blood. Now the blood's ionic balance got little disturbed. So because the ionic balance got little disturbed, now the blood will give back everything to the back to the tubules. That will go as your waste. Okay, that will go as your waste. Clear? Yes or no? This is Java class. Arc boy Java. Okay, next C++ C classes. Yes. Done. Shall I continue? Yes. Okay. Very good. Okay. So students, as I told you, in this, the main thing that is going to happen is during your urine formation, the blood, okay, the ionic balance has to be maintained. Okay. Has to be maintained. To maintain this ionic balance, sometimes we do secretion. Okay. Sometimes we do secretions. That could be secretions of K plus ions, H plus ions, okay, it could be HCO3 ions, all these ions, no, ammonia, mainly ammonia, okay, ammonia ions, all these ions we will be secreting back to the tubules, okay, we will be secreting back to the tubules. I think it's very easy to understand, yes or no? So this is a very, very important step for maintaining the ionic balance and also the osmolarity of the entire filtrate, okay. So this step is mainly for that. Now you can look at a diagram here. Can you all just look at the diagram and understand? This one is called as your peritubular capillaries, okay, peritubular capillaries. So this kind of diagrams will help you imagine the things that you cannot imagine, right? Otherwise, all this is just told. Okay, all this is told. Now you'll be able to understand, okay, capillaries are there, tubules are there. Okay, I'm able to relate something, right? So look here, students, what is going to happen? Some things which I don't want in my tubules, that will go back to the peritubular capillaries. At the same time, whatever the blood does not require, it will give it back. Now, example, sodium will be given to the blood. Okay, sodium exchange is going to happen from the tubules to the blood. From the capillaries, what is going to come? K plus, okay, NH3, H plus, all this will come from the tubules here. Let's do one thing. Let's just write it in a different color. Okay, let's say whatever is coming from the blood, K plus, NH3, okay, H plus, it's coming. So automatically what is going to happen? Here K plus you can see, okay, here H plus you can see, automatically it got into the tubules, okay, NH3, these things got into the tubules. Are you able to imagine? Yes? Able to imagine? Yes or no? Tell me fast. Yes, okay, very good, very good. So these are the main things that is going to happen during the tubular. So to maintain the osmotic balance in the blood, okay, to maintain osmotic balance, tubular secretion will take place. So these are your tubules. I'm labeling this also. This is your peritubular capillaries. Yes, fine. Now I told you about what are the main 3 steps of, all of you tell me what are the main 3 steps of urine formation in the chats, all of you quickly. By then I'll draw the PCT. Okay, by then I'll draw the PCT. Till that, all of you tell me. Yes, quickly answer. Which are the 3 steps of your urine formation? Quickly in the chats. Waiting for your answers. Okay. Now, look here. Very good. Yes. Ultrafiltration, tubular reabsorption, tubular secretion. I hope all the technique is clear. Pressure you understood. How much filtrate is going out you understood. How much blood is going out. You also understood which is the pressures that is helping them to move inside and what is the total pressure. Then you also understood from tubules to your blood how reabsorption is happening, how secretion is happening with amazing diagrams. Now you are going to understand how the tubules are going to individually help in this whole process. Okay, individually going to help in this whole process. Now students, understand this is your PCT. Inside your PCT, what is there? Brush border epithelium is there. Cuboidal simple brush border epithelium is there. So let's draw this. Oh no. Now students, this has very less diameter for me to draw this. Give me 1 minute. Okay. This is your PCT, okay, large structure I am drawing. Yes. Inside this, who is going to be there? Your brush border epithelium, right? Brush border, cuboidal brush border epithelium. This is how the cells will be there. Yes, ma'am, this is how the cells will be there. Now, what is this PCT exactly going to do? What is it going to reabsorb? What is it going to do is the biggest question, okay? Now, reabsorption will happen, students. Reabsorption of many things will happen, okay? Reabsorption of many things like your NaCl, okay, glucose, water, HCO3 minus nutrients, amino acids. All this reabsorption is going to happen. All this reabsorption is going to happen. Can you all think of it? Yes or no? Yes, can you all think of it? Yes, ma'am. Okay, all this reabsorption is going to happen. Many things are there. Okay, electrolytes, urea, some amount of urea for countercurrent mechanism. All this reabsorption is going to happen. Okay, all this reabsorption is going to happen. Done? Dusted? Clear? So, your glucose, NaCl, H2O, all this active absorption is going to happen. Okay, all this active absorption is going to happen. See, your nitrogenous waste, all this, your passive absorption is going to happen. This I had already mentioned. Yes or no? Yes, ma'am, already mentioned. Now, students, understand, other than urea, your Cl is there to maintain your osmotic balance. So this people, let's say these people, okay, NaCl up to your electrolyte will get reabsorbed actively. Okay, actively. But at the same time, your nitrogenous waste will get absorbed passively. Okay, passively. So PCT is going to do, very important thing, PCT is going to do 70 to 80% of reabsorption. I have added NCERT SNP also, so you will not have any doubt. Okay, 70 to 80% reabsorption is going to take place because of your PCT. So PCT is the one who is going to majorly do the reabsorption. PCT is going to do active, passive, reabsorption will continuously happen. Around 70 to 80% reabsorption will happen because of PCT. Any doubts in this? We are also going to put NCERT and learn it. Okay, NCERT and learn it. Done and dusted? All of you in the chats? I cannot see your chats there. Okay, all of you in the chats? Okay, so maximum reabsorption is happening where? Here, right? Maximum reabsorption. This is all NCERT line, okay? This is all NCERT line. Maximum reabsorption is happening. Any doubts, please ask me in the chats. Okay, here potassium ion also we have to put. Okay, potassium is very important. Sodium, potassium. Where to put potassium, students? Okay, potassium ions. So maximum reabsorption has happened. Yes, ma'am, maximum reabsorption has happened because of PCT. And which is the cell this is present, students? Which is this? They are going to help in increasing the surface area. Okay, they are going to help in increasing the surface area. Is my voice still heard? Yeah. 1 second. Who is that in the chat? Yes, okay, very good. So these cells are your simple cuboidal brush border epithelium. Okay, brush border epithelium. Done? Okay, ma'am, understood. PCT does 70 to 80% reabsorption. Maximum reabsorption occurs in the PCT. Now, let's see your NCRT, what NCRT is telling. Proximal convoluted tubule or your PCT is lined by simple cuboidal brush border epithelium. Yes, ma'am, understood. Which can, what is the main job? To increase the surface area for reabsorption. Nearly all of the essential nutrients, that is around 70 to 80% of the electrolytes and water are reabsorbed by this segment. Okay, 70% of electrolytes and water. Now, PCT also helps to maintain the pH and the ionic balance. Please do understand, your entire tubules will help in maintaining the ionic balance. Okay, ionic balance of the body fluids by selective secretion of hydrogen ions, okay, hydrogen ions, ammonia and potassium ions into the filtrate and absorption of HCO3 from it. Okay, they will take HCO3 from it and they will give, whom and all they will give? H plus hydrogen ions, ammonia and potassium. Ammonia and potassium. Who is disturbing, yeah? Yes, gone. Okay. Okay. So, absorption will happen. You should tell me. You should tell me. Don't tell them. Tell me. So, hydrogen ions, ammonia, potassium ions into the filtrate. Okay, and by absorption of HCO3 from it. Okay, now loop of Henle, reabsorption is minimum in its ascending lift. However, this region plays a significant role in maintenance of a high osmolarity of medullary interstitial fluid. This you are going to understand when I teach you countercurrent mechanism. Countercurrent mechanism. These descending limb of loop of Henle, okay, is permeable to water but almost impermeable to electrolyte. So, descendant means descending. Okay, descendant means what? A person who is going to come next. My descendant is going to be this next teacher. That means after me, or like I am going to give all my power to this teacher. So, descending means downwards, ascending means upwards. I will show you the limbs, don't worry. Now, this concentrates the filtrate as it moves downwards. The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, the concentration of the filtrate will happen. Are we ready? Shall we draw loop of Henle? Okay, let's draw loop of Henle, students. Okay, proper U-shaped structure. Proper U-shaped structure. Yes or no? Yes, this is your loop of Henle. Now we have to do little shading. So, in this, if you understand only, you will understand countercurrent mechanism. So, listen carefully to the class. Don't talk to each other. Listen carefully. Very important topic to understand countercurrent mechanism. Okay, this is your loop of Henle. Now, what are they telling? They are telling this loop of Henle have two limbs, ascending and descending. Okay, ascending and descending. Oh God. One second. Okay, ascending and descending, very important. Now, which is ascending, ma'am, which is descending? For that, you have to understand, students, when you're coming down, okay, descendant. So, this is going to be, Okay, this is going to be, let's draw it this way, descending loop of Henle. This portion is going to be descending loop of Henle. This portion is going to be ascending loop of Henle. Now, loop of Henle, minimum reabsorption happens, that is minimum reabsorption happens in the ascending loop of Henle is what they told. Now, what are they telling? They are telling that this people mainly helps in osmolarity, okay, to maintain the osmolarity. Let's say, okay, this is one portion, this is one portion. Students, between the loop of Henle and the vasa recta, there is an intermedullary. So, this portion will be your medullary interstitium, okay. Medullary interstitium will be present here. See, after this, this is your cortex, okay, this is your cortex. So, from the cortex, when they are coming out, the osmolarity is going to be around, in cortex it is going to be around 300. But as it comes down, okay, as it comes down, it is going to go from 400 to 600, 600 to 800, 800 to 1000, 1000 to 1200. How is this happening? Okay, how is there an increase in osmolarity is how you are going to understand. Look here, descending loop of Henle is permeable to water. Okay, descending loop of Henle is permeable to water. Ascending loop of Henle is impermeable to water. So, here what are they going to do? All of you in the chats. What is happening? Tamil, you don't have to study, dear. No one is forcing you to study. Okay? Yes. So, here what is going to happen? Water is going to come. Yes, water is going to come. And this is permeable to water. Okay? Can you see blue color, students? Little dark, no? One second. Give me 1 minute. So, water is coming. This is permeable to water. Say H2O. Okay, H2O, H2O. Water will. Now, understand, when water is continuously oozing out, inside the concentration of the filtrate will become hypertonic. Agreed or disagreed? Okay, in this water, imagine in this water there was lot of sugar. Okay, after some time I start removing the water. Then I am going to get an exact sugar liquid, right? Exact sugar liquid. That is because all the water is removed, the electrolyte concentration is very high. In that condition, we call it hypertonic solution. So, students, as it is coming here, you can see osmolarity has reached up to 1200. Yes, ma'am, it's very visible. Okay, very understandable. Yes, ma'am, osmolarity has increased because all the water is getting drained out, electrolyte concentration is increasing. By the time it reaches here, your osmolarity, that is your concentration, is going to be around 1200. This is going to be the osmolarity here. And this osmolarity is not just created like that, it is created because of the presence of NaCl and urea. I will teach you in countercurrent mechanism. Now, students, look here, water is coming. Here, what did they tell? Here they told that ascending lymph is impermeable to water. It will not bother if water is there or not. It will be bothered about electrolyte. So, here electrolyte, NaCl. Okay, here everywhere NaCl is going to come out. No water will be reabsorbed. So, this side what is going to happen? Hypotonic will happen. This side hypertonic will happen. Is this clear? Yes or no? Any doubts? Please ask me right now. Fast. Okay. So, students, here NaCl, here NaCl. Now, there is a catch here. According to your NCERT, there is a catch here. This is going to be thick part of your tubule. Can you all see how thick it is? I have drawn it thick, okay. This is going to be thick part of your ascending loop of Henle. This is going to be your thin portion. This is going to be a thin portion. Now, in the thin portion, okay, there is some differences. Okay, there is some differences. In thin portion, students, the absorption, the reabsorption, okay, will happen. The reabsorption will happen passively. Passively. This question has come in PYQ, okay. Passively it will happen. In the thin part of your ascending loop of Henle, passively absorption will happen. In the thick part of your ascending loop of Henle, actively it will happen. actively it will happen. Okay, this PYQ has come actively and passively. So very clear, loop of Henle is there. Okay, in the loop of Henle, what is happening? The descending loop is I'll write it here for all of you. It is permeable to water. Okay, permeable to water. Students, all these notes you will get it on Vedantu Neet English Telegram as well as on my Telegram, that is Biology by Gopika. Okay, so if you want to join the Telegram, you are always welcome to join the Telegrams. So this is permeable to water. This one is impermeable to water. Okay, impermeable to water but permeable to salts or electrolytes. Okay, impermeable to water. Okay, understood. Now automatically here water will get removed out. Okay, water will go. Here the electrolyte concentration is going to be max to max. Okay, electrolyte concentration is going to be very, very high. Electrolyte concentration is going to be very, very high. Now I told you that osmolarity, something called as osmolarity is there. In the cortex is going to be 300, but as it comes down it will go 400, 600, 800, 1000 to 1200. Why? Because you can understand here the solution is going to become more hypertonic, and this is not just happening because of that, it is also happening because of your NaCl as well as your urea. Okay, now as it comes here, your osmolarity has reached up to 1200. Okay, the medullary interstitium concentration has completely increased. Now when you go up, student, the ascending loop of Henle is impermeable to water. So it is going to not bother about water, it is going to, but it is permeable to the electrolyte. So all the NaCl will start moving out. Now in the ascending loop of Henle, there are two segments, thin and thick segment. Thin segment, what is going to happen? NaCl passively it is going to get reabsorbed. In the thick filament, it is going to get actively reabsorbed. So understand this side, in your descending loop of Henle, here the solution is going to be hypertonic. In the ascending loop of Henle, it's going to be your hypotonic. Okay, ascending loop of Henle, it is going to be hypotonic. Let's write it down that also. This here, in this region, the solution, okay, here the filtrate is going to be hypotonic. Okay, hypotonic. Here it is going to be hypertonic, this portion. Okay, hypertonic. This is going to be hypertonic. Okay. Now NCERT told one more statement, that is, in the thin segment of your ascending loop of Henle, very minimum reabsorption happens. Okay, minimum reabsorption happens. In loop of Henle itself, minimum reabsorption only will happen. Okay, lot of reabsorption will not happen. Mainly they are going to be there to maintain the osmotic balance. Okay, osmolarity will be maintained by your loop of Henle. Is this clear? Yes or no? Yes, perfect. Okay, fine. This is your loop of Henle. Okay, this is your loop of Henle. Now students, here, now that you understood this, okay, let's read now once again this. Henle's loop, reabsorption is minimum in its ascending loop. However, this region plays a significant role in maintenance of high osmolarity of your medullary interstitial fluid. The descending loop, okay, the descending limb or loop is permeable to water but impermeable to salts, that is electrolytes. This concentrates the filtrate as it moves downward. Imagine 300 to, we came up to 1000, 1200, right? The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrates pass upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Can you understand this? As it is coming downwards, it's going to get hypertonic, concentration is going to increase. As it moves upwards, it's going to become hypotonic because water is not getting reabsorbed. Hypotonic, only Na will be moving. So minimum reabsorption will happen in the ALOH. Okay, minimum reabsorption will happen in your ascending loop of Henle. Minimum reabsorption happens in the ascending loop of Henle. I hope it is clear. Okay, there cannot be a better diagram to explain you this. Now, the next thing is DCT. Distal convoluted tubule, conditional reabsorption of Na and water takes place in the segments. DCT is also capable of reabsorption of HCO3 and selective secretion of hydrogen and potassium ions and NH3 to maintain the pH and sodium potassium balance. So what is DCT going to do, students? It is going to do conditional reabsorption of Na and water. That means when necessity is there, I will do the reabsorption. If necessity is not there, I will not do the reabsorption. Conditional reabsorption is done. Then DCT is also capable of reabsorption of HCO3 also and selective secretion of hydrogen and potassium ion and ammonia to maintain the sodium potassium this pump. Clear? Okay. Then collecting duct. This is a long duct extends from the cortex to the cortex of the kidney to the inner parts of the medulla. The large amount of water could be reabsorbed from this region to produce a concentrated urine. Again, depending on when the situation is there. Okay. This segment allows passage of small amounts of urea into the medullary interstitium. Very important. Countercurrent mechanism, you will understand this. Okay, to keep up the osmolarity. It also plays a role in maintenance of pH, ionic balance of the blood by selective secretion of H plus and potassium ions. Okay, potassium ions. So, what did they tell for DCT? They told DCT can do conditional, okay, conditional reabsorption of Na and water, and it can also do conditional reabsorption, that is it can do reabsorption of HCO3 minus, and it can do selective secretion. Very important, okay? Selective secretion of H plus, K plus and ammonia. Main job of DCT, PCT, everyone is to maintain ionic balance, okay? Maintain ionic balance and also the pH, okay? pH, clear? Clear? What is happening? All of you are hungry? I'll give you a break. Chill people. First you study, okay? After that I'll give you break. I'll give you after that. Yes. So if I draw PCT here, right, DCT, sorry, if I draw DCT here, how is it going to be? We'll write this down, no students? I don't think I can copy paste here, so I'll write this down. Okay, if you draw DCT, this is how your DCT is going to look. Okay. This is how your DCT is going to look. What are they going to give out? It is going to give out NaCl, H2O, HCO3 and selective secretion of NH3, K plus. Yeah, K plus, H plus. Got it? This is how your DCT is going to look. DCT. Okay, diagram also done. Okay, diagram also done. Okay, so I'll write here to maintain. Okay, this is all to maintain the and pH. Okay, maintain the ionic balance and pH. Done, done. Done. Okay, next we'll go to collecting duct. Collecting duct, again, lot is not there, students. Collecting duct, you already read it, right? What is that? It is basically this collecting duct also, let's say from here, This is the collecting duct. Collecting duct also can reabsorb water. Okay, it can also do reabsorption of water, and it can also do to maintain the osmolarity. To maintain the osmolarity, it will give out urea, right? These are the things that they told. What else they told? One more point they had told. All of you tell me, thinking, tell me what is one more point. Yes, the reabsorb water I wrote. The main job of it is to do anyone? It will do mainly maintenance of pH and ionic balance by, very important, okay, by selective secretion H plus and K plus. Yes or no? This is what they told. Agreed? Disagreed? Okay? Yes? So, what are they going to do? They are going to give out urea. Okay, that urea is going to go into your PCT and your DCT. They can also do selective, that is H plus, okay, and your K plus. This is what they have told. Okay, very easy, your collecting duct is very good. Yes, increase the medullary interstitium. Yes, totally, absolutely right. Do you want a break now? After this, I'll give you a break. See, look here, all of you. So, students, here we saw in PCT HCO3, NaCl, H2O, nutrients, K plus, H plus, NH3, all this we saw. Then when we came down the lane, we saw descending loop where it will take only water. As we came down, again we saw ascending loop where there is a thin segment and a thick segment. In the thin segment, actively or passively it will be happening. Thick segment actively. As we go up, we can see in the DCT NaCl, H2O, HCO3, K plus, H plus, and then you have collecting duct. In the collecting duct, all of you notice here, urea is going here and it is getting into the loop of Henle. This will help you understand the countercurrent mechanism. Okay, this is the diagram in your NCRT and this is the full overview of you whatever I taught you still now. Any doubts, please do ask me now. Okay, any doubts, please ask me now. Any doubts? No, ma'am. Clear. Okay. Do you guys want a lunch break? Are you all hungry? Okay. Are you all hungry? I'll give you a break. And after that, we have mechanisms and then disorders. Okay. After this, maybe in 1 more hour class. So, shall we take a lunch break? Okay. Even I'll go eat something and come. So, okay, I'll also eat lunch and come. See you all. Okay. Yes, even I'm hungry a little. So, we'll see at 2:10. After that, we have only 1 hour class. Okay. Not even 1 hour, maybe 45 minutes. Okay. Done. Come back. Okay.
Its mic is on now.
That's it.
It's so much of glare here, sometimes I can't even see. Hello students, let me know in the chat if you're able to hear me. Hello, hello, hello. Yes. Students, let me know how the class is going on right now, because Gopika ma'am is telling me it is going fabulous, but the problem is you're not sharing the sessions. That's what I'm hearing a complaint about from Gopika ma'am, that you're not sharing the sessions, you're not bringing your more friends to the channel. Students, understand at this time when there is so much of time left, all of you will be thinking, sir, why should I be serious right now? Students, if you're not serious right now, it's going to be very difficult. And I want everyone to bring their own friends here so that all of you can have like, entire like group where all of you can study and everything. Right?
Exactly.
So this is, and where is attendance, ma'am?
No attendance also, sir. I think scolding, we'll become old.
Yeah, exactly. We'll only become cold. I'm saying cold. Cold also is also old. So.
Yeah.
Yes.
Okay, Srinetra has come from DNA. Break time.
Break time.
Yeah,
DNA
okay.
is over today. Actually, no more class.
Oh, no more class. Then I expect all of them to join.
Yeah.
Sir, scold your DNA students, ask them to join. Let me teach some...
Students, join here because you need to study at least around 4 to 5 hours per day. That is minimum. So if you're doing that 4 to 5 hours with us and self-study of 2 hours, you're good to go from now onwards.
Exactly.
So...
Yes.
Yes, ma'am.
So students, all of you please mark your attendance and let's continue the class. We have some mechanism, disorder, then we're done. Okay?
Mechanism.
Yeah. RAS mechanism.
RAS mechanism.
Yeah.
RAS one, RAS one.
RAS.
Bye, ma'am.
Bye-bye. Yes, ready? All of you? Okay, before I go to RAS mechanism, I'll do countercurrent mechanism. Okay, after countercurrent mechanism, I'll do RAS mechanism. So by the time all of you just fresh up and get back to the class, I'll be drawing the diagram. Okay, I'll be drawing the diagram for you all. So this is your tubule. Okay, let's make okay, ready? Yes. So students, quickly smash the like button and we are going to continue. So this is countercurrent mechanism. Now ma'am, why do we need countercurrent mechanism? Understand students, during some conditions, like normally when we are at a lot of, say when we are fasting, when we are not having food properly, our body goes through a stress condition. So during the stress condition, the osmolarity will automatically drop. That means the ionic balance of the body will drop. It's because we wouldn't have drank water, we wouldn't have had enough salts in our body. So to adjust to this, our body has something called as countercurrent mechanism. Now from the name, what do you understand? Countercurrent means when two tubes are in close proximity with each other, and in this tube the fluid is going in this direction and in this tube it is going in the opposite direction. This is called as countercurrent. Okay? Counter, all of you know, when you go to like some talk shows and all, they'll tell something, you will give back answer to that. That is called as giving counters. Okay, good point of counters we give. Same way, when two tubes are in close proximity and when both the liquids are flowing in opposite direction. Now, which are the two tubes? All of you can look at it and tell me. This is the loop of Henle and this is your vasa recta. Now, all of you go back to our first ever image. this image. In this image, can you see yellow color is the tubule? Can you see vasa recta? So this portion I have exactly drawn like that. This portion will cause the countercurrent mechanism. Okay, so I've taken a small portion of this and I have pasted it here. The pasted one is this. Okay, the pasted one is this. Now, ma'am, why are you teaching about countercurrent mechanism? Because it's very important for you to understand between whom and whom it's happening and what is actually happening. How are they increasing the osmolarity? That is what we are going to understand. So, students, this is down one. Always I told you, down one is going to be descending loop of Henle. Okay, this is going to be ascending loop of Henle. Done? Ascending loop of Henle. If here it is descending, then obviously vasa recta is going to be ascending. Ascending vasa recta. Okay, ascending vasa recta. And this is, if here it is ascending, here it is going to be descending vasa recta. Then who is this? This is your CD or your collecting duct. Now, they both are in opposite direction. Yes, ma'am, I agree. These two are in opposite direction, these two are in opposite direction. Now, how the things are going to happen, right? How the things are going to happen also you need to understand. Ready? All of you want to draw? I'll give you 2 minutes if you want to draw. If you don't want to draw, I'll continue. Yes, Devika, I'm from Kerala. I am born in Kerala, brought up in Bangalore, so I am a mix, hybrid I am. Okay, hybrid. Okay, chalo, start. Okay, now fluid or your filtrate will be flowing like this in your descending loop of Henle, like this. Okay, we just saw this now. It will be going like this and it will come up this way. Now, if it is going this way, then your blood is going to flow in the opposite direction, obviously. So, it is going to go upwards. Okay, go upwards. So, that's when we call it countercurrent. See, one is going that way, the other one will go this way. This way. Okay, now how does it happen? Students, it mainly happens because of your NaCl and urea. It mainly happens because of your NaCl and urea. We already know in your descending loop of Henle, right, in your descending loop of Henle, all the water molecules is going to come out because DOH is permeable to water. In the ascending loop of Henle, all the electrolyte is going to come out. Now, which is the electrolyte I drew? I drew NaCl. Yes or no? I drew NaCl. So, NaCl is going to come. And between the loop of Henle and vasa recta, I told you people, there is an interstitium. Okay, there is a fluid that is called as medullary interstitium. Let's say this is the fluid. Okay, this is the medullary interstitium. Now, we have to concentrate this to concentrate the urine. Okay, let's say the body is under stress condition. We need to make sure that there is no more loss of water. So, we are going to concentrate the urine. How is this going to happen? Now, NaCl came out of your ascending loop of Henle. This NaCl will immediately be taken by your descending vasa recta. Okay, exchange they are going to do. Exchange they are going to do. Whatever is coming out of your descending, that is ascending loop of Henle, will be taken up by your vasa recta. Vasa recta will carry it all the way like this downwards. Okay, all the way like this to the, up to the descending vasa recta, and drop your NaCl in this interstitium. Okay, drop your NaCl in this medullary interstitium. Got it? Medullary interstitium. Now, students, understand this NaCl concentration is more in the medullary interstitium. Okay, medullary interstitium, this is more. Okay, concentration, we are making the solution hypertonic solution. Okay, hypertonic solution. Now, once it became hypertonic solution, collecting duct is like, wait a minute, I have some urea also, I can give you that urea. So, from here, the urea will go all the way into the medullary interstitium. Okay, this urea will go to the medullary interstitium, and now here urea also will be there. Okay, urea also will be there. Now, automatically, this medullary interstitium has become concentrated. Yes or no? All of you agree? Now, here students, the medullary interstitial fluid, okay, the medullary interstitium fluid has become hypertonic. Hypertonic. How it became hypertonic? With the help of NaCl and urea. Now it is very understood that from the tubules all the water will start coming out, right? All the water will start coming out. Okay, water will start coming out. Yes or no? When I made the solution hypertonic, obviously the water is going to come out. This way the urine is going to become more concentrated. Urine is going to become more concentrated. Is this clear to all of you, countercurrent mechanism? Yes or no? Okay. So, urine concentration we are increasing. More than the main thought is not to increase the urine concentration. It is to save as much as water possible, so that if this person has not consumed during stress conditions, especially in your juxtamedullary nephron, this kind of mechanisms happen. So, students, basically 3 countercurrent is happening, right? Between this two, between this two, between this two and between this two, right? All are opposite. Just see all the opposites. Okay, all the opposites. So this is what is going to happen. This is called as countercurrent mechanism. So what is going to happen basically? First thing, we are going to increase the osmolarity. Okay, osmolarity. So I already taught you in loop of Henle, right? When I taught you loop of Henle, see, look here, I showed you the osmolarity. It's going from 300 up to 1200. So where is it happening? In the medullary interstitium. Now same thing I'm going to show here. Okay, from here, let's say from here, if it was 300 in the cortex, okay, by the time it reaches here, it is going to be 1200, okay, osmolarity. Okay, osmolarity up to 1200. So it starts from 300 up to 1200 will be the osmolarity. Okay, so osmolarity will increase from 300 to 1200 osmol. Okay, osmol. Then the second thing is NaCl and urea will increase the concentration in the medullary interstitium. Medullary interstitium. Okay, that is, they will make this hypertonic. Hypertonic. Now automatically what is going to happen? Okay, automatically what is going to happen? Urine is going to get extremely concentrated. Okay, extremely concentrated. Urine is going to get extremely concentrated by the removal of water. By the removal of water. Any doubts? Any doubts? Please ask me right now. Hi Srinath. Yes, 4 times more concentrated, exactly. 300 to this one. Okay, clear? So now you saw this one, that is your loop of Henle. So this will be more clear for you, right? How it is going from here to here. Okay, how it is going from here to here to here. Now students, one diagram is given in your NCERT, that also I'll move it up. Yeah, this is the diagram. Okay, look at this diagram. I have drawn it in a more easier way to understand, but here if you see, from 300 it will go up to 1200. Okay, so here they have done, this is your thick loop of Henle, that is ascending loop, and then this is thin one. So you can see how it is happening. Okay, Na will go out, that Na will be taken by a vasa recta, it will pass it all the way, drop it in the medullary interstitium, and it will continue. From the collecting duct, urea, can you see urea? Urea will go to the medullary interstitium and concentration. This is a more complicated diagram, and it will not be easy to make you understand the concept with this diagram. So I drew the diagram and then I am showing you this diagram. Okay, so this way you will be able to understand, okay, NaCl is coming, it's going to the vasa recta, their concentration is increasing. Same way urea also is coming, because of which reabsorption of water is happening everywhere. Can you see? Reabsorption of water is happening. This way the urine will get automatically concentrated. Understood? So from cortex to medulla, they will ask you what is the increase in osmolarity. You have to tell 300 to 1200. Done, done, done. Okay, done, done, done. We'll go to the next mechanism. Next mechanism, yes or no? Drawing Bowman's capsule, this board also is showing me where you should draw. What is telling you? Draw here. Okay. Now students, RAS mechanism. You already know this mechanism. Ma'am, what are you telling us? I taught you JGA apparatus. Yes or no? I taught you JGA apparatus. I told you that whenever there is a problem with the GFR, okay, whenever there is an issue with the GFR, the issues can be different, students. Say blood volume is low, okay, blood volume is low, or say Na is less, okay, Na is less, because of which BP is low, GFR rate is less, okay, the amount of GFR that has come in is less. Multiple problems can be there. These are the problems. Now we have to find solution for this. How is the solution happening? That is very simple, that in the afferent arteriole, some of the smooth cells will get modified to form JG cells, okay, modified to form JG cells. On whose signal they will become JG cells? This JG cells is formed, okay, JG cells is formed from the signal sent by macula densa, okay, signal of macula densa which is present in the DCT, okay, macula densa of the DCT. After that, this JG cells is going to release something called as renin. This renin cannot do anything alone. So what renin will do? He will be like, okay, I cannot do much, but whatever possible for me to do, I will do. Okay, I hope all of you smash the like button. Let's make it 100 likes, all of you, quickly. This renin will go and convert, okay, will go and convert angiotensinogen. Okay, students, let's draw. Okay, it will convert angiotensinogen released by the liver. Okay, released by the liver, but it will be in the plasma. Okay, let's say this is the plasma, okay, in the blood vessel. Angiotensinogen released in the blood vessel, okay, by formed by the liver, okay, formed by liver, will be released here. This renin will go convert angiotensinogen, okay, it will convert angiotensinogen to angiotensin 1. Angiotensin 1. But this angiotensin also is in an inactive stage. Not completely inactive, but little, okay. This is completely inactive. Angiotensin 1 also will be like, wait, I cannot fix any issues. So angiotensin 1 will convert to angiotensin 2. But how does this happen? It happens with the help of our one and only, in breathing and exchange we studied, your one and only lungs. Okay. Lungs will be like, don't worry, I have a solution for you. I will release, okay, I will release ACE. What will it release? ACE, which is called as angiotensin converting enzyme. It will release angiotensin converting enzyme and it will allow it to form angiotensin 2. Angiotensin 2. So, what basically happened? Here JG apparatus happened, okay, JG apparatus was happening. Okay, this is your entire JGA apparatus. This, if you people did not understand, you had to watch the first part of this lecture, okay, lecture 1. JG apparatus, this JG cells release renin, okay, release renin. This renin told, okay, wait, I cannot fix the issue by myself, so what I will do is, I'll go convert angiotensinogen, which is present in the plasma formed by the liver. It is inactive stage, okay. I will convert angiotensinogen to angiotensin 1. We were like, okay, fine. But this angiotensin 1 also is not in a fully active state. So, angiotensin 1 will be like, okay, wait, I will convert myself to angiotensin 2 with the help of lungs to release ACE, that is angiotensin converting enzyme. Now, this angiotensin 2 have the ability to fix all this issue. Any doubts so far, students? Any doubts so far? angiotensin converting enzyme. Any doubt, please ask me. ACE enzyme. Now this angiotensin 2 will be like, okay, wait, what is the issue? Okay, sodium is less. Let me do something. I will activate, okay, or I will send signal, okay, or angiotensin 2 will basically stimulate your adrenal cortex, okay. It will stimulate adrenal cortex to release aldosterone. This aldosterone will do the job of conditional reabsorption. Okay, it will do the job of conditional reabsorption of Na plus. Okay, it will do conditional reabsorption of Na plus. First problem sorted? Yes, ma'am. See, Na plus automatically increase, so automatically BP also will increase in volume. Next one was problem with the GFR. So angiotensin 2 will be like, okay, don't worry, I have another solution for that. I will do vasoconstriction. Okay, I'll do vasoconstriction. Okay, I'll do vasoconstriction of your EA, that is your efferent arteriole. Why is he doing that? Simple thing, I told you that time also. If here it is more and here it is less, then more blood will come in, so more GFR. Okay, more GFR. So he'll do, okay, vasoconstriction of EA, I'll do, automatically GFR will increase in size. Now the next problem is water. Water is not there. So they'll be like, okay, let's do reabsorption of water also. We can again tell DCT to do, or we'll tell collecting duct to do, but we can get it done. It's not a big job. Okay, it's not a big job. So the next task is to do reabsorption of H2. So reabsorption of H2 will fix this issue of blood volume and water low. Clear or not, students? So angiotensin 2 will do the entire job of fixing all those issues, because if these issues exist, then your urine formation also will become a problem. Got it? This is called as your RAS mechanism. Clear? So once again, let's do a quick recap. JJ apparatus was there. This JJ apparatus released renin. Renin was not able to do everything alone. So renin went and converted angiotensinogen, which was formed by the liver, which was present in the plasma. Okay, which was present in the plasma, to angiotensin 1. Angiotensin 1 also was not able to do the whole job. So angiotensin 1 converted to angiotensin 2 with the help of ACE enzyme, that is angiotensin converting enzyme, by lungs. Now angiotensin 2 will be like, don't worry, I can fix all the issue. Okay, that is stimulate adrenal cortex to release aldosterone. That aldosterone will do conditional reabsorption of sodium. Automatically the blood pressure and the sodium level will be balanced. Next thing is reabsorption of H2O. They'll be like, okay, you do one thing, do reabsorption of water. That way the blood volume also will be fixed. Okay, blood volume also will be fixed. Next thing was, I will do vasoconstriction of efferent arterioles. Vasoconstriction of efferent arterioles will make sure that the GFR is, right, GFR rate is increased, because efferent will become bigger, efferent will become little smaller. Got it? Yes or no? Is this clear? So he told, okay, I'll do vasoconstriction. So this is how vasoconstriction will happen. This is how it is. Okay, lumen is there. And like this contraction will happen. So this is what the whole system is. Yes. Anyone has doubt with RAS mechanism? So whenever GFR has an issue, that is your glomerular filtrate rate has any issue, say it including itself, like no more, less is there, any kind of issue, everything will be fixed by your RAS mechanism. Okay, done. Next is ADH mechanism. Okay, ADH mechanism. This is called as anti-diuretic hormone. All of you know the hormone or not? Yes, please tell me in the chats. Please tell me in the chats. Fast, done. Any doubts? Ask me now. After this, you will not be able to ask the doubts. So, ask right now. So, everything is pakka clear. Okay? Pakka clear. Okay. Okay, students. So, antidiuretic hormone is simply called as, okay, let's write the full form first. Antidiuretic hormone or vasopressin. Okay, vasopressin. Students, here you have to understand, this is when your hypothalamus, okay, hypothalamus will send your neurosecretory cells to the posterior, okay, posterior pituitary, and your posterior pituitary will only release two hormones, that is vasopressin and oxytocin. This posterior pituitary will release vasopressin and oxytocin. All of you know this, right? Now, that is your ADH. Now, why is this mechanism there, ma'am? What is the actual problem? The problem is diuresis, loss of water. We have to prevent or get in more water, right? Loss of water means a lot of water is getting lost. We have to take back the water. So, ADH will be released. Is this clear? Now, what I have to do? I have to somehow make sure that the water is getting saved. One thing that ADH can do is, that is vasopressin will do is, it will do exogenous intake of water. It will tell us that, okay, we are thirsty, now take in some water. Okay, exogenous intake of H2. This will be happening. Second thing that it can do is telling our kidney for reabsorption, right? Kidney reabsorption. That means reabsorb more water. Reabsorb more water. Possible? Possible? Yes, possible. Okay. That is kidney reabsorption. That is basically fluid reabsorption will happen. Okay. Fluid reabsorption. So, this will automatically, students, this fluid reabsorption will automatically increase, okay, the blood volume, that is the water content in the blood, okay, blood volume. Yes or no? So, the problem is that this person is losing a lot of water, okay. It could be happening because of diabetes mellitus, that means sugar is more, right? Loss of water. Loss of water. This we have to save by first exogenous intake of water, that is giving that thirst, okay, thirst mechanism, that means telling that, okay, you are thirsty, please drink water. Second thing is we can do fluid reabsorption. This way automatically the blood volume will increase, yes or no? Yes. Maybe one more thing that we can do is blood vessel constriction, that means we increasing the more GFR to come, more lack of loss of water. Okay, lack of loss of water. These two things we can do. Yes or no? So, conditional reabsorption we can do exogenous intake of water. Any doubts in this? It's a very simple thing. Okay, very simple thing. Students, and this also, loss of water happens. Because of loss of water, automatically osmolarity also will become less. Okay, osmolarity also will become less than 300. So, we have to make sure that the water is saved. Okay, water is left back. So, to fix this issue, only this can happen. Okay, so automatically because of these two things that is happening, the urine will get automatically concentrated. Okay, urine will get automatically concentrated. This way we will be able to save the water that was before getting lost. So, this is urine will get concentrated. Done. ADH mechanism is just talking about the hormone. Okay, vasopressin is there. It will basically make sure that the loss of water will be taken care. Automatically, because of the loss of water, osmolarity also will automatically drop, right? So, here hypothalamus will release this into the posterior pituitary, that is your ADH or your vasopressin. That will do, first it will give an telling you take in water. Thirst, exogenous intake of water. Second thing is kidney reabsorption or fluid reabsorption, which will automatically increase the blood volume. Third one also I have read some places, that is constriction, okay, constriction of your blood vessel. But I think it is not mentioned in your NCERT. Okay, yes, done. Yes. So students, the blood vessel constriction part is just to increase the pressure, atrial pressure. So more will come in, no? So that is the main job. Okay, if you want, I'll write it somewhere. Where do I write it? Okay, wait, I'll erase this and I'll write it here. Okay, that is blood vessel constriction. This will automatically increase the atrial pressure. Okay, atrial pressure will automatically get increased. This way more, the negative pressure also can be little cancelled out, because of which urine will get concentrated. Okay, only RAS mechanism is a little continuation or a little bigger mechanism. Other two mechanisms are very small. Okay, very, very small. Done. Any doubt, ask me right now. Can I continue? Okay, fine. Okay. So next is ANF mechanism. Okay, this is called as atrial. Okay, atrial natriuretic factor. This is actually done by the heart. This is actually done by the heart. Okay, atrial natriuretic factor. So in this one, if I say exactly, this fellow is a check. Okay, he is going to check on RAS mechanism. Check on RAS mechanism. Because if there is a no check on RAS mechanism, while fixing all the issues, they might also lead to a new issue. Okay, because GFR rate is getting increased or something else is getting increased. So because of it, there is a check. There should be a check, you know. Oh, are you becoming too much? Wait, I will break your bones, no, like that. So to check on RAS mechanism, ANF mechanism is there. It's actually against RAS mechanism. Like RAS is basically, let's say, protecting from loss of water, protecting from NaCl absorption and all that. This person will do ulta. Okay, it will do two things. Main thing it does is it will do diuresis. It will do diuresis, that is loss of water, and it will do natriuresis, uresis. So this is basically whenever water is more, it will make sure that there will be water will be removed out. Okay, and it will also do the most important thing, it will do vasodilation. RAS mechanism was doing what, students? Vasoconstriction, right? This will do vasodilation. So this fellow is actually ulta of RAS mechanism. This one will make sure that there is loss of water when the water is more. Okay, loss of water. So the problems can be what? The problems can be, let's say, blood volume is high. Okay, that means too much water in the blood. Blood volume is high. H2O is high. NaCl is high. Okay, Na plus ions are more. Let's say BP is extremely high. In these situations, ANF will actually act upon. Okay, ANF will actually act upon. That is, loss of water will be done. At the same time, you know, loss of your sodium ions. Okay, loss of Na plus ions. And it will also do vasodilation, that is dilation of the blood vessel. Okay, dilation of the blood vessels, then the GFR rate that is coming in also will be decreased. Okay, so this is actually, in other words, we can tell they are antagonistic in nature. Okay, antagonistic to RAS mechanism. Yes or no, students? It's doing opposite of RAS, no? Do you agree or no? It's doing opposite to RAS, right? Any doubt in this, anyone? All of you in the chats? No, right? Okay, fine. Clear? Yes. Here it is going to do vasodilation. RAS was doing vasoconstriction. So it is actually a check on RAS mechanism. Ki, okay, are you reabsorbing a lot, or maybe are you doing too much of conditioned reabsorption? That kind of things will be fixed by this fellow. Okay, that kind of things will be fixed by this fellow. Okay, agreed? Chalo, let's go to the next topic. Micturition. My students already know this. So if urination should happen, right? If urination should happen, then what should happen, students? So let's say this is your urinary bladder, okay? There is this trigon of bladder and all that, right? Let's say, oops. Okay, let's say urine is filled inside this. Now the urine has to come out, right? Urine has to come out through the urethra. For that to come out, there are going to be, first thing, on the urinary bladder there is going to be stretch receptor. Okay, on the urinary bladder there is going to be stretch receptor. This stretch receptor will send signal to the CNS, central nervous system, telling that, okay, I am full. Now you do one thing, you actually contract. That contraction will be done with the help of a detrusor muscle. Okay, detrusor muscles will contract, and this detrusor muscle will also allow the urethral sphincters are there, no? That is, your internal sphincters will relax. Okay, internal sphincters will relax. Then external sphincter is in our control. So then we'll also get a signal that, okay, you have to use the washroom. So external sphincters also will relax, and the urine will, micturition will happen. Okay, micturition will happen. Very simple, no? Stretch receptors are present on the urinary bladder. Okay, let's say it's present here. This stretch receptors will send CNS, okay, will send signal to your brain. Okay, to your brain. This brain will send signal back telling that, okay, you can do contraction, and automatically your detrusor muscle will contract, your sphincters which are present also will relax, and because of which the urine will come out. Is this clear? Urine will come out. Very simple, right? Yes, perfect. Detrusor muscles. Yes, all of you understood very well. Okay. So students, here understand motor neuron, okay, from here to here signals, here to here signal, that you don't have to take tension, but just understand this is your entire signaling. Now as I told you, most important thing is your GFR that will go out in per minute. It is going to be 125 ml per minute, okay, per minute, and or else you can say 180 liters per day, okay, per day, and out of this only 1 to 1.5 liters of urine will be given out. Okay, 1 to 1.5 liter of urine only will be given out. Done. This is what you have to know. Done. Any doubts? Ask me. Okay. Yes. Yes, done. Okay. Now students, in the urine, the color in the urine is because of your urochrome. Okay, because of your urochrome, the color, right? One straw yellow we can tell, right? One straw yellow color will be there, and depending on seeing your urine color, the doctor can actually understand what might be the issue in your body. Okay, what might be the issue. Now in your urine, if there is a lot of glucose, okay, glucose content is high in your urine, then you will have a condition called as gluconuria. Okay, glucose. And if in your body ketone bodies, okay, ketone bodies are more, then you will have ketonuria. Okay, ketonuria. Got it, all of you? Any doubts? Okay, glycosuria and ketonuria. Okay, yes, these are the two conditions. Very, very important. I'll put it in a box for all of you. Yes, done. Okay, very important words. Glucose is more in your urine, glucosuria. Ketone is more, ketonuria. Okay, glucose in urine. Should I write it here? People will understand, no? Is more. Okay, done. Now we are going to understand. What are we going to understand? Yes. Students, other than my kidney, other organs also are giving out some excretory substances. So let's see. Okay, let's see what are those. See, our lungs. Okay, our lungs remove large amount of carbon dioxide. We already studied in breathing and exchange. Approximately 200 ml per minute, and also significant quantities of water every day. Okay, liver, very, very important. Liver, the largest gland in our body, secretes bile containing substances like bilirubin, pigments. Okay, bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, drugs. Most of these substances ultimately pass out along with the digestive waste. So that means your fecal matter, it will go out. Okay, now most of this substance will go out. Now the sweat and the sebaceous glands in the skin can eliminate certain substances through their secretions. Sweat produced by the sweat glands is watery and it is salty. All of you know, your sweat is salty. At least while running you would have, okay, taken in some. So small amounts of urea, lactic acids also will be given out. So in your sweat, what and all will be there? NaCl will be there, some amount of urea will be there, some amount of lactic acids will be there. Though the primary function of sweat is to facilitate a cooling effect of the body, it also helps in removal of nitrogenous waste. Now, sebaceous glands eliminate, sebaceous glands is your oil glands, they eliminate certain substances like sterols, hydrocarbons and waxes through sebum. Okay, they'll remove all this through sebum. This secretion provides a protective oily covering for the skin. Do you know the small amount is given out through saliva as well? Saliva as well. Okay. Diabetes mellitus is when your glucose level is more. Okay, glucose level is more. Understand that. This you will study, I think diabetes mellitus and insipidus you will study in chemical coordination. Okay, chemical coordination you will study. Got it, all of you? What are they saying? Ma'am, which is diabetes? Students, diabetes mellitus is when your glucose level is very high in the body. Okay, even when you have that, you have a tendency of drinking a lot of water, doing very frequent urination. Now, diabetic insipidus is when you have a salt and water. That means your osmoregulation is not happening properly in your body. So, diabetes insipidus will be that time condition. Okay, diabetes mellitus also they say a lot of symptoms are similar only, but then the diabetes insipidus is caused when your salt and water balance is broken. Okay, got it, all of you? Yes. So that is when normally we tell diabetes, oh you have diabetes, but it is actually you have diabetes insipidus. Okay, diabetes mellitus is only sugar content. Diabetes insipidus is when you want to always drink water, you want to always frequent urination. That is the difference. Understood, Sony? Yeah. So I told by mistakenly diabetes mellitus is diabetes insipidus. Okay, insipidus. Okay, done. Is this clear? Very simple. Okay, this one just remember your liver. Bilirubin, biliverdin, cholesterol, all this will be given out by steroids will be given out by your liver. Then you have lungs, carbon dioxide and mainly some water. Mainly carbon dioxide, some amount of water. Then you have sebaceous glands which will give out your oil, sterol, hydrocarbons. Sweat glands will give out NaCl, lactic acid and your urea. Okay, these things you have to understand. Now we will do disorders. All of you tell me, just now we are doing some disorders, right? We just discussed some disorders. What else? Now there is a renal failure. That is the first ever disorder. One second, students, this mic and I am dancing with together. Okay, first ever thing is renal failure. Renal failure simply means kidney failure. So when a renal failure happens, a kidney failure happens, we will go for dialysis. Now if one kidney fails, another kidney will automatically function. All of you know this, no? Even with one kidney, a person can happily live if that person is living a healthy life. Now both the kidneys are failed, the person will go for dialysis. Or say if both the kidneys are there, one kidney completely failed, one kidney has got some infection, they'll go for dialysis. So kidney failure, you have to do dialyzing or dialysis, right? Now what is dialysis, ma'am? Very simple, students. If I have to teach you in a very simple way, let's take this as your, there is a cellophane tape that will be there, okay? There is going to be a cellophane tape that will be there. Now if I put this, sorry. 1 second, I'll connect, first I'll connect this mic properly. Can you still hear me, all of you? Yes? Okay, you can all hear me, right? Okay. Now, look here. This is a cellophane tape. What tape? Cellophane. Okay. Cellophane tape. These tape will have some pores. Okay, I am telling how to do hemodialysis. Okay, hemodialysis. This will have some pores. Now, what is the whole thing? This cellophane tapes, we will have the blood, okay, from the person who has had a kidney failure. The blood will be there, okay, this blood will be there. Students, this blood is rich, okay, it is rich in urea. That means it is rich in urea means uremia is there. So when urea is extremely more in your blood, you will have a condition called as uremia. Okay, you will have a condition called as uremia when your urea is extremely high in your blood. Now this blood has uremia, the blood has urea. Now when I am using a dialyzing fluid, let's say this is your dialyzing fluid. This is your dialyzing fluid. I've put, this is just to make you understand, okay, this is not how it will look. This dialyzing fluid is absent of urea. This dialyzing fluid, I had made sure that this dialyzing fluid, dialyzing fluid is absent, okay, no urea at all, absence of urea or your nitrogenous waste. Automatically what will happen? Urea will move from a region of higher concentration to a lower concentration. So from this blood, the urea will automatically start coming to this dialyzing fluid. Agreed, all of you? This is normal mathematics, normal biology diffusion. So this blood was rich in urea. Okay, blood was rich in urea because his kidney has failed. So no one is there to do the excretion. Since the kidney failed and it is rich in urea, obviously the urea will automatically start moving into the dialyzing fluid because the dialyzing fluid does not have any amount of nitrogenous waste. So automatically urea is moved from a region of higher concentration to a region of lower concentration. Now when I am giving, okay, when I am taking the blood from this person, I am going to give heparin for this. Okay, I am going to add heparin. Why? Because I have to make sure that the blood does not clot. Okay, when I am giving this purified blood, okay, let's say, students, oxygenated, deoxygenated, don't get confused. Okay, oxygenated blood will have nitrogenous waste. Okay, oxygenated, deoxygenated means just in the terms of your presence and absence of oxygen. Okay, oxygen. Understood? Oxygenated blood will be toxic blood but with full of oxygen. Deoxygenated blood will be full of carbon dioxide but no toxic, no nitrogenous waste. So now when I give this blood back to the person, I am going to give that person anti-heparin because the body already has heparin. Okay, body has naturally heparin. So I will give anti-heparin for this person. This is all you have to know in hemodialysis. So when urea content is more, we call it as uremia, right? When urea content is more, we call it as uremia. And in dialysis, it's very simple. We take a cellophane tape. The cellophane tape will have pores because exchange has to happen. Okay, exchange has to happen. So this pores will allow the urea exchange to happen from a region of higher concentration to a region of lower concentration. That is the person's blood. Okay, the person's blood. Now when I'm taking that person's blood, I will give it heparin so that coagulation does not happen, and when I'm giving back the blood to the person, I will give him anti-heparin, because blood, the body already has its automatic, like natural heparin done. Is this clear to all of you? This is called as hemodialysis. Okay, this is called as hemodialysis. Hemodialysis. But is this a permanent solution? No, this is not a permanent solution. This is a temporary solution. And also understand, this is a lot of financial crunch, because dialyzing every month or every 3 months, 6 months, depending on that person's condition, is a very expensive process. Okay, so a person will not be able to take it. So this is not a permanent solution. This is a temporary solution. Permanent solution is going to be a kidney transplantation. Okay, that is going to be kidney transplantation. Now in kidney transplantation, what is the big problem? They will tell, see, mostly it is preferred that you take from your relatives or someone very close to your family, like from your family or from your relatives. Okay, this way graft rejection, right, graft rejection is reduced. Okay, graft rejection can be reduced if you take it from your relatives or your family members. Understood, students? Any doubt? Okay, understood? Okay. And yes, we can give cyclosporin A, that is an immunosuppressant, so that graft rejection does not happen. But kidney transplantation is a permanent solution. This is a temporary solution. Okay, this is temporary solution for a kidney failure, and this is a permanent solution. Okay, permanent solution. Now we have some more diseases. Let's see that also. First one is renal failure. See, a functional kidney is used in transplantation from the donor, and preferably a close relative, to minimize chances of rejection by immune system of the host. Modern clinic procedures have increased this chances. Now renal calculi, that is stone or insoluble mass of crystallized salt, especially your oxalates. Okay, oxalates. This oxalates, what will happen, students? Now imagine this is your tubules, right, to the kidney. It will come and get stuck here, because of which filtration will not happen properly. Filtration will not happen properly. Yes. So this renal calculi, that is your simply called as kidney stone. That is when oxalates gets accumulated in your body, and because of it they will form stone shaped structure. Normally for 90% of the people, they will tell drink a lot of water, or they will give some tablets, and that person will be fine. In a very few situations, they will go for a complete surgery or an operation. Okay, that is when the calculi becomes very big, the stone becomes very big. Okay, and they know that a tablet cannot help. Now next one is glomerulonephritis, that is when inflammation of the glomerulus of the kidney. So the glomerulus is there, where your capillaries are there. When inflammation of that happens, understand, if that once inflammation happens, your entire GFR will become a problem, your entire filtration also will become a problem. Okay, understood? Got it? This one is your glomeruli of the kidney, that is glomerulonephritis, inflammation of the glomeruli of the kidney. Did we miss anything, students? Just check your NCRT. I'll also check my NCRT once. We did hemodialysis, renal calculi, glomerulus, diabetic insipidus. We don't have, no? We don't have diabetes insipidus, right? Everything is pakka? Yes? Just check everywhere. Okay, just check. Did we miss something? Did we? After that, I'll make you do some questions. Okay, after that, I'll make you do some questions. Yes, chapter dandana done. Exactly. Chapter is dandana done. Yes, anything? Anything we left? Check everywhere. Anything? Nothing, no? We missed something. We missed something. One thing, that is your pathway of urine, that I think I taught in the first class actually. Okay, It's okay, no problem. It's just based on how it's coming from collecting duct, duct of Bellini, minor, major calyx, renal pelvis, ureter, urinary bladder. That is what I wanted to tell. I think I had told when I taught you the diagram. Okay. Something else did we miss? Nothing, I guess. Yes, nothing. Okay, so this dialyzing, students, dialyzing is like your artificial kidney which will do the job of your kidney. Shall we start? Okay, let's do the questions. So before we do the questions, I want to let you all know that there is a lot of offers happening in our VLC centers, especially for students who have scored 600 plus. You get a complete 100% scholarship. That means without paying any money, you can join the offline center and continue your preparation. This is something that we want to help a lot of students also, because students who have scored 600 plus are definitely work day and night to get that marks, but because of the rank inflation, people who are say up to 660, okay, or let's say up to 655 also might not get into their desired dream college. Okay, their desired dream college. Because of that, we are giving them a complete 100% scholarship. Go to any VLC center, show your Neet score, show your scorecard, and they will give you an entire coaching for completely free. Other students, these are the prices you can see in Pune, Delhi, Patna and AP. So if you have any doubts, please do feel free to contact this number and join the offline centers if there is a VLC center near your place. Okay, yes. So shall we do the first question, students? Can you all see the question? Okay, yes. Statement 1 says that in the nephron, the descending loop of Henle is impermeable to water and permeable to electrolytes. Statement 2, the proximal convoluted tubule is lined by simple columnar brush border epithelium and increases the surface area for reabsorption. So they are telling descending loop is impermeable to water and permeable to electrolytes. And this one they are telling proximal convoluted tubule is formed by columnar brush border epithelium. Please tell me the answer, all of you. Yes, quickly. Okay, Hasini has told the answer. I want more answers in the chat. Yes. Naina 3. Why Naina 2 is correct? Savita, check once again. Students, proximal convoluted tubule is lined by columnar or cuboidal? Check properly your NCERT and tell me. I'll wait for your answers. Proximal is with cuboidal or columnar? I think it's cuboidal. What do you guys think? Yes, both statement 1 and 2 are false, because this is ascending, or let's say if it is descending, it is permeable. Okay, proximal is called cuboidal, right? Cuboidal columnar epithelium. Next one, choose the correct statement given below regarding juxtamedullary nephron. Stress, okay, when you are in stress. So please read, students. My energy is all gone. Okay, read and tell me the answer. Let me see the question. Renal corpuscles of juxtamedullary nephron lies in the outer portion of the renal medulla. Loop of Henle of juxtamedullary nephron runs deep into the medulla. Choose the correct statement, okay? Yes. B is the answer? Yes, perfect. Loop of Henle of juxtamedullary runs deep into the medulla. Perfect, perfect. Next question. Okay. Which part of the human nephron is impermeable to water? Impermeable to water. Okay. Yes. Impermeable to water. Students, 24. Need 2024 questions. Okay. Sony, focus here. People will come and go. You have to continue your preparation, okay? Next year more rank inflation happens also, you should be in a position to get into a good government college. Okay. Muhammad is writing shairi, song he is writing. Yes, C is the answer, ascending. Okay, ascending lib. Very good. Next one. Given below are two statements, one is labeled assertion, other is labeled reason. Juxtaglomerular apparatus play an important role in regulation of the GFR. Reason, a fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby bringing GFR to normal level. Okay, this is the question. I hope all of you can read it. Juxtaglomerular apparatus play an important role in regulation of the GFR. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby bringing GFR to normal level. This is the question. Yes, A is the answer. Let me see. Perfect. Yes, very good, very good. That means you people have understood the chapter well. If you are able to solve the question, that means you have gained something out of the class, right? When you are able to answer, please do understand, that means your concepts are clear. You know exactly what to answer. Okay? So students, this comes when you sit in a class where you understand every single topic. Otherwise, you will not be able to do the questions. Okay? Fine. Next question. Again, two assertion reason. Nephrons are two types. Okay? Cortical and juxtamedullary. 2023 question. Okay? All PYQs. Based on their relative position in the cortex and medulla. Reason, juxtamedullary nephron have short loop of Henle, whereas cortical nephrons have longer loop of Henle. Okay, assertion reason, which is true or false. Yes, try. I hope it's visible. Check the assertion reasoning properly, okay? A is false, R is true, both A and R are true, but R correctly explains A. Okay, both A and R are true, but R does not correctly explain. A is true, R is false. Very good. A is true, R is false, because this should have been ulta. Cortical nephron has short loop, juxtamedullary have long loop. Okay. Next question. Arrange the events of renin-angiotensin mechanism in correct sequence. Let's see how many of the students understood. Activation of JG cells and release renin. Angiotensin 2 activated release of aldosterone. Fall in glomerular blood pressure. Reabsorption of Na and water from the distal convoluted tubule. Angiotensinogen is converted to angiotensin 1 and then angiotensin 2. Okay. First let's do the order, then we will do the option. Okay, which is the first one, students? Let's go like this. Tell me which is the first one. Where do I put number 1? After that, you can do the, what is that? Problem. Like you can solve this. First, tell me which is first one. Tell. C is 1. C is 1. That is fall in the glomerular blood pressure. After that, activation of JG cells to release renin. Yes or no? After that, this one. 3. That is angiotensin is converted to angiotensin 1. Then 4. Then 5. So it is going to be option number 1. Yes or no? Perfect. Option number 1, C. Okay, very good. You people are telling, okay, C. Yes, this is correct. Order is correct. First problem should come. Then solution will come. Then release will happen. And then this, then this. Yes, A is the answer. Very good. Shall I give you homework question? Yes, here you go. Your homework question, Neet 2022 question. Which one of the following hormone reduces the blood pressure? reduces the blood pressure? Antidiuretic hormone, atrial natriuretic factor, aldosterone, angiotensin 2. Okay, angiotensin 2. This is the question, which one will reduce the blood pressure? 1, 2, 3, 4. Which one? You can give the short forms in the chat box. Okay, you can give the short forms in the chat box. I hope everything was clear, students, today. Okay, so do not forget to let me know in the chats if you really like the session and if you understood everything and you are able to do the questions as well. And thank you so much for joining. See you all. Make your decision wisely if you want to join offline or online. Till yesterday we had our DNA offer and a lot of our students have joined there as well. So see you all soon with the next class. Next class I think I'll do evolution. After that I'll do locomotion. Okay, because I'll take 12th, 11th both the syllabus together simultaneously. So both will get over, right? So yes, we will do evolution next because I love evolution. It's a storytelling. Okay, it's like a class where I can talk a lot. So yes, students, thank you so much, and definitely let me know if you like the session, and also let me know in the chats how the session was. Thank you so much for joining. All these notes will be available on the Telegram within 5 minutes. And yes, that's it. Thank you so much. Tata, bye-bye. Don't forget to put your homeworks also in the comments, okay? Yes, love you too, Hasini. Yes, love you all. Okay, bye-bye.